something strange about this babysitter. She called the blood bank and asked if they deliver. She walks by the mirror, but I don't see her reflection. I've seen Twilight. Believe me, it left an impression.
And everybody knows they come to suck your blood.
Hey!
The guys who look their eyes, you got nowhere to run.
Hey! Her fingernails are sharp, her teeth are pointed too. Mom, let me alone with her. Now what am I gonna do? The babysitter's a vampire. She's looking at me like I'm dinner tonight. Stay away from my neck. The babysitter's a vampire. Gonna need a wooden stake and a garlic milkshake to keep me alive. Oh great, my mom left me alone with a vampire. A bloodsucker.
Hello, today our group is going to be presenting to you on Draculin, as will be further discussed by my lovely teammates. Draculin, yes, being named after the classic 1800s novel Monster, is a glycoprotein which inhibits the coagulation pathway, specifically coagulation factors 9 and 10. This is theorized by researchers such as Wara L. Francesca to be an adaptation of the vampire bat, in order to ease the consumption of their prey's blood. Due to interest concerning this anticoagulant, research has been done to explore Draculin's medicinal properties. This protein was in fact found to be a potential treatment as a blood thinner and general aid for heart problem victims, as can be found in studies such as the vampire venom research of Dr. Kakumano et al. Draculin has also been shown to be greatly beneficial for ischemic stroke victims, which accounts for 87% of all strokes according to the CDC. This is especially crucial function since there is a high demand for less dangerous and less time-sensitive treatment for stroke victims. Currently, the most popular post-stroke treatment is TPA, or tissue plasminogen activator, which works in breaking down fibrin-tangled blood clots. After a stroke occurs, if the clot forms in the penumbra, the cells are potentially salvageable as long as the blood flow and vital oxygen are restored promptly. However, TPA itself induces neuronal toxicity and heightens neuronal death when not administered within 3 hours of the stroke. More specific ill effects of TPA can be seen in the figure shown. This was taken from the tissue plasminogen activator study. This is a problem since, again, according to the CDC, only 38% of reported stroke victims were aware of their symptoms and thus able to seek medical attention within those 3 hours. After which TPA many times is not given to the patients since the cons do not outweigh the pros. Failure to get medical attention from a stroke can cause brain damage, long-term disability, and death. An enzyme, though, DSPA, which is produced by Draculin, when introduced into the bloodstream causes this breaking down of blood clots, which proved beneficial for stroke victims, and it did not carry the same neuronal toxicity properties as TPA. In Reddrop et al.'s research, in order to test the effect of DSPA, Dr. Reddrop and her colleagues injected mice with NMDA, that's N-methyl D-aspartate, in order to induce a stroke-like lesion. Then, after 24 hours, the mice were split into 2 groups, one receiving TPA and the other receiving DSPA. The results show damage and cell death caused by NMDA was only aggravated by TPA, whereas no additional toxicity was seen in DSPA. In similar studies, this lack of toxicity had made researchers such as Robert L. Metcalf, in his article, theorized that this could be injected up to 9 hours after a stroke without any increased neuronal damage. This is substantial since stroke victims are currently extremely time-sensitive patients, and this therefore could triple the amount of time a patient could seek treatment. One of the only main downsides of Draculin is in obtaining it. In Fernandez AZ et al. Study, they discovered that the anticoagulant properties of Draculin decreased if saliva from vampire bats was collected daily, only recovering if given a 4-day resting period. By observing the difference in high and low activity saliva, they found significant decreases in composition of the carbohydrate moiety and glycosylation pattern. Furthermore, controlled chemical deglycosylation of native Draculin under conditions that do not affect the polypeptide backbone progressively led to complete loss of biological activity. In other words, the study's results implied that correct glycosylation of Draculin is a seminal event for the expression of the biological activity of this glycoprotein. However, when this is all said and done, Draculin could potentially significantly improve stroke victims' lives, decreasing risk in treatment and allowing the availability of more people to seek treatment.
Draculin is found in the saliva of vampire bats. The vampire bat will bite its victim and secrete the enzyme Draculin into the bloodstream of its victim to prevent the blood from clotting. So right now we know of 3 species of vampire bats, the D. rotundus, the D. youngi, and the D. caudata, which all contain different forms of DSPA, which is another name for Draculin. And Draculin is a non-venom form of a plasminogen activator, and it has 5 different domains. And those 5 domains are fibronectin type 1, EGF-like, K1 and K2, and the serine protease. So the D. caudata, which mostly feeds on birds, contains all of the 5 domains in its DSPA. The D. youngi and the D. rotundus, they both feed on mammals, but the D. youngi's DSPA lacks the K2 domain, and the D. rotundus's DSPA lacks the K2 domain along with the plasmin-sensitive activation site. So the one that we'll be focusing on is the common vampire bat D. rotundus, because it's the only one of the 3 which has been used for different research, especially in aiding stroke victims, because of its anticoagulant properties. So the 2 other species, D. youngi and D. caudata, do produce DSPA, but their Draculin doesn't have the same anticoagulant properties, and therefore isn't as adept in breaking down blood clots. So all 3 of these species do contain an anticoagulant because they all drink blood, and they've adapted to allow for them to continue drinking the blood without it clotting, which would stop them from feeding. So right now, Draculin is competing in the market with TPA, which is a blood thinner that has been used to treat stroke victims. And their structures are similar, as you can see in this picture. So on the top you have TPA, and on the bottom you have DSPA. And you can see in the box, highlighted in blue, where the K2 domain has been deleted from DSPA, but TPA still has that K2 domain. And the yellow triangle shows where the plasmin-sensitive cleavage site has been lost as well. And they believe that the deletion of the K2 domain in Draculin seems to make it more fibrin-specific, and also lowers the probability of side effects, along with the plasmin-sensitive cleavage site being lost. And now some fast facts about Draculin. It is a glycoprotein that has 441 amino acids and weighs about 88.5 kilodaltons. It was also discovered in 1998, which is the same year that I was born. It was first tested on humans in 2006, where the testing showed that it was safe for humans. And it was given the name Draculin, which named after Count Dracula, because the enzyme does come from vampire bats. And my citations are an article called Dracula's Children, Molecular Evolution of Vampire Bat Venom by Dulce H.W. Lowe, another article called Desmoplace, Discovery, Insights, and Opportunities for Ischemic Stroke by Robert L. Medcalf, a website called Drug Bank, another article called Structural Biology and Protein Engineering of Thrombolytics by Jan Mikav, and then an article called Draculin, Stroke Drug from Vampire Bats Moves Closer to Circulation by Patrick Morgan.
Isn't Draculin just fascinating? But how is it made, and what is the pathway? From what I could find, it goes through translation in a ribosome like any other protein, with tRNA and codons and making all the amino acids to make our protein of Draculin. But the structure that we can get is very interesting. But after translation, it's glycosylated and made into a glycoprotein. As Grace had mentioned before, it cannot function properly without this step. Johanna mentioned that there are different DSPA isomers in different vampire bat species. One such isomer is missing a K2 domain and a plasmid-sensitive activation site. Now, the missing domains are very important in how it works. The tissue plasminogen activator, or TPA, is a protein that we naturally make in our bodies to help break down blood clots. This TPA is very similar to DSPA in how they do that. They both activate plasminogen to plasmin to tear down the fibrin in a blood clot. But TPA has all 5 of the domains that Johanna has mentioned, and it would not be able to function properly without these domains. So how is it that these molecules can do similar things with different structures? It has been found that though DSPA has no K2 domain, it does have a K domain. Now, this domain is structurally similar to the K1 domain in TPA, but functionally it works like a K2 domain. This is also how DSPA avoids inhibition. The plasminogen activator inhibitor 1, or PAI1, is what binds the TPA to inactivate it. It cannot do the same, and it doesn't do it as fast to DSPA without that K2 domain. Without the plasmin site, DSPA can be active in a single chain form, unlike TPA, which is active in a 2-chain form. And it cannot be regulated like TPA since it works primarily as a venom. TPA is regulated based on the amount of fibrin in the body. As the amount of fibrin goes up, TPA becomes activated and works on plasminogen, but as the amount of fibrin decreases, it is inhibited and it is inactivated. Now we can see how TPA activates plasminogen to become plasmin to break down fibrin in this diagram, but we can also see how it can be inhibited by the PAI1. But DSPA doesn't care about these mortal regulations. It hates fibrin with a passion. It's so sensitive to fibrin that it obliterates any it can find by activating plasminogen and prevents it from synthesizing by inhibiting factors 10 and 9 in the coagulation pathway. Now, DSPA can keep a cow bleeding for hours after a bite. The glycosylation is the key. It is what helps it remain for so long. When it's glycosylated at the same points that TPA is, but DSPA is glycosylated with a highly bioprocessed N-glycan, while TPA is higher in mannose in its glycosylation. Now, this difference for DSPA means that it is cleared at a 4-fold lower rate than TPA, resulting in a very prolonged activity time. This is all so that a vampire bat can feed for as long as it needs. Now, the venom particularly comes from a submaxillary gland in the bat, and once a bat feeds and releases its venom, it takes approximately 4 days for that gland to refill. From this, I hypothesize that DSPA is regulated in the bat based on the amount of DSPA in Draculin that the venom already has. And since it takes about 4 days, it is probably a little slow to make, but since it's made along with the other components of the venom, that can be understandable.
So looking closer at how Draculin is produced in the bat has epithelial cells, and inside the nucleus of this epithelial cells we'll find the genome. Somewhere in this genome we'll find what encodes for the Draculin protein. And so this Draculin gene is going to be transcribed into messenger RNA and then translated into a protein. So as the messenger RNA is post-transcriptionally modified in the nucleus, it'll exit the nuclear pore, it will enter the cytoplasm. Initiation factors will help it find the small subunit on the ribosome, the 40S subunit, and then this will bind with the messenger RNA. It'll travel along to the start codon, it'll start translating. As the large subunit combines, it'll form the ribosome and start producing this protein. Once Draculin has left the ribosome, it will travel through the epithelial cell where by the glands it will be injected into the wound, and when the saliva comes in contact with the wound, it will inhibit the coagulation pathway through the inhibition of factor 9 and 10, and then it will activate the fibrinolytic pathway through the conversion of plasmin to plasminogen. So this will prevent the coagulation of the wound as well as help to dissolve any existing blood clots. So in order to understand how Draculin is interacting physiologically, we need to understand both the coagulation pathway as well as the fibrinolytic pathway. So with the coagulation pathway, we can either start intrinsically, where contact activation from surface damage will activate intrinsic pathway, or we can start extrinsically after tissue trauma releases tissue factor. Either way, both these pathways are going to lead to a common point where they both activate factor 10, and then activated factor 10 will convert prothrombin to thrombin. And then in turn, thrombin is going to convert fibrinogen to fibrin. So once these fibrin monomers begin to develop, they will polymerize into covalently linked fibrin fibers, and then these fibers will, under the presence of activated factor 13, they're going to develop into a fibrin mesh that attracts platelets and phospholipids and forms a clot. The fibrinolytic system is composed of 3 serine proteases. This is plasmin, tissue plasminogen activator, TPA, and urokinase plasminogen activator. So these 3, along with Draculin, these are all going to be serine proteases, and they're going to work on the activation of plasminogen to plasmin. So this is important in terms of dissolving physiological clots for wound healing, as well as prevention from forming a clot where we don't want them. But when plasmin is produced and present in the blood, it's going to degrade all these blood plasma proteins, including the fibrin clots. And so this is going to degrade the fibrin clots. With regards to how our protein is broken down and eliminated, the protein is going to be hydrolyzed into amino acids, and then from here it can be used to aid further biosynthesis, or it can be further processed in oxidative deamination, and we'll get a amine as well as a carbon skeleton. The amine will be sent to the urea cycle and either reused or excreted via urea, and then the carbon skeleton, depending on whether or not it's glucogenic or ketogenic, the glucogenic amino acids are going to be broken down into pyruvate, used for gluconeogenesis, or sent into acetyl-CoA, just like the ketogenic amino acids. And they're sent to acetyl-CoA, where they can either be broken down into fatty acids and ketone bodies, or enter the citric acid cycle and electron transport chain for further energy production. So looking closer at the mechanism for our transamination, we're going to see our alpha amino acid here is going to react with our enzyme-linked PLP shift base, and so that's drawn here. We're going to see the alpha amino group of the amino acid being transferred to PLP. This is going to give us a geminal diamine intermediate, and then yield an amino acid PLP shift base called an aldamine. And then our enzyme linked amine will fall off. And that's that.